Hello, everyone. My name is Logan Cohen, and I'm a senior in biology at Worcester State University. Today, I'm going to be discussing the annotation and comparative analysis of insulin-like peptides and their interacting genes across Drosophila species. Before I begin, I'd like to give special thanks to my faculty advisor, Dr. Darren Barnard, and also to the Genomics Education Partnership for providing the materials that are necessary to complete this research. What we're going to go over today is a little bit of scientific background because this presentation is intended for a general audience. So if you're not a science major, that's all right. I got you. We're going to go on a little bit of a tour of the insulin signaling network, and then we're going to come up with some discussion points and potential questions for future research. Now, in order to understand this, you need to understand cell signaling. Cell signaling is the way by which cells respond to their environment. The way that this happens is that these receptors pick up on signaling molecules that are called ligands in the environment. The receptor then sends a signal into the cell and initiates pretty much a game of telephone, where the message is relayed to the next signal peptide and the next signal peptide until it ends in its intended target and you end up with a cellular response. Now, ILPs are very interesting. ILP stands for insulin-like peptide. If you think about insulin in humans, we immediately think about diabetes, but insulin does so much more than that. Insulin-like peptides in Drosophila control things like sleep-wake cycle, adiposity, gut health, brain function, reproductive function, and transition from one life stage into the next. Now, because of all of these critical functions, you would expect this gene to be very well conserved, which means it would be very similar in one species of fruit fly to the next, right? Well, that's not what I found. When I went through and I did a comparative analysis of ILP6 across 4 different species of Drosophila, I found an enormous amount of change, more than it's expected in such a well-conserved network as the insulin signaling network. So that brought up a question. Well, how far do these changes go, right? Because you can have a difference in the ligand, but that doesn't mean that you have a change in the expression further into the network or into the cell. Now, INR and SDR are the 2 receptors for insulin-like peptides in Drosophila. INR is the receptor that picks up a signal and sends it off to the relay module, or the relay peptide. And so this is the one that encourages expression. SDR is another receptor, but it's different than INR in that even though it has the same binding site, it doesn't send the signal off to the inside of the cell. So what this means is that we have 2 different genes, one that upregulates expression and one that downregulates expression. Now, what I found was pretty predictable. There is a difference in INR and SDR across the phylogeny that does seem to be relative to the amount of change in the ILP genes themselves. But what about going onto the inside of the cell? Looking at our first relay receptor, that would be Chico. Chico is the peptide that picks up the signal from INR and sends it off to the inside of the cell, setting off that cascading game of telephone. What I found was actually pretty incredible. It's in the 2 different subgenuses that I was looking at, Chico has 2 completely different sections of the genes called exons between the Drosophila subgenus and the Drosophila sophophora subgenus. This is really interesting because I wasn't expecting to find that much of a difference once we got to the inside of the cell, but there it was. So I decided to go on to the next genes in the cascade. Now, what I ultimately found, looking at over a dozen different genes, is that yes, the changes in the conservation, or better put, the changes in these genes between species did extend into a depth of the network that I did not expect. And an example of that would be IMPL2 and ILP2. Now, these 2 genes work together. Remember, ILPs, those are our ligands, attaches into INR, goes through Chico, which sets off the game of telephone, and then after a few different other receptors, we end up at IMPL2. Now, what does IMPL2 do? It modulates growth. So IMPL2 itself had an enormous amount of change relative to the rest of the conservation in the network. And I thought that was really interesting because when we look at the difference between species, when we look at their phenotype or their external appearance, we do see a lot of differences between, let's say, species like Drosophila hydae, which is in the Drosophila subgenus, and Drosophila melanogaster, which is in the Drosophila sophophora subgenus. We see that hydae has a body size that is twice the size of melanogaster. It also has an extended lifespan relative to melanogaster, with a lifespan reaching up to 25 days compared to melanogaster's average of 15 days. There's also a lot of differences in gametic expression, so expression in the reproductive system of reproductive cells, and other metabolic differences that do seem to be products of these changes in the insulin-like peptides. So what we're seeing is not only that the ligands are different or in between species, but also that the final product that's set off by these ligands does also seem to be different. And that brought me to this question: is the evolutionary paradigm incomplete? Now, what do I mean by that? I mean that gradual and random changes to the genome over time are, we know, to be a mechanism of evolution. But are there more concerted and adaptive means of evolution? So what I mean by this is, do ILPs drive evolution in Drosophila species? Are they a loci of evolution? Are they a trigger gene? And does the variety of expression and the variety of ILPs available in Drosophila provide this concerted mechanism for evolution? Now, I don't have at this point the information to actually go and dig into that question, but the questions that I would ask are: how could we explain the depth of change in an otherwise well-conserved network? Because the insulin signaling network, on the whole, is extremely well conserved, not just in Drosophila, but across the entire animal kingdom. So this is a network of genes where we don't expect to see a lot of change at all, and yet here we are. Another question would be, how are ILPs expressed in Drosophila? Can you express ILPs under condition of shock or stress? And would these expressions produce an inheritable change in the organism? And if we find that to be true, are there other well-conserved networks in Drosophila or in other species that provide the same sort of mechanism for evolution? So these are all questions that I would love to dig into. But right now, I'm really looking forward to your questions at the conference. I hope to see you all there.